Hi, everyone. This is the first lecture for week 7 on gene regulation for microbiology course. So these slides are on Camino, so you can download them and follow along. So the first question is, how do we control gene expression, or maybe more importantly, why do cells control gene expression? And the bottom line here is that most microorganisms live in environments that are dynamic. They change. So they don't need all of the gene products that they could potentially make at all times. There are often products that are only useful under certain conditions. At the same time, there are gene products that are basically need all the time. So the gene products that can be on at some level all the time are called constitutive genes, right? So again, these are genes whose products are basically needed at some level all the time. This might include things like ribosomal subunits or DNA polymerase encoding genes, RNA polymerase encoding genes, etc. So things that carry out very basic functions in the cell, the ability to make lipids, things like that. Whereas other examples might be sets of genes that are only required to metabolize a certain product from the environment when that product is around. The example we're going to talk about today is the Lacl. So you could control gene expression at the level of controlling transcription or translation, or even after translation. But usually this is done at the level of controlling the initiation of transcription, right? So here we have DNA being transcribed to make RNA, and this requires the binding of RNA polymerase to the promoter and the initiation of transcription. So that's the most common site for regulation to happen. But you could also control how stable the messenger RNA is. You could control the ability of the ribosome to translate the messenger RNA. And then after that, of course, there's a lot of things that can control the activity of a particular protein or enzyme, or the stability of that protein or enzyme. Right, so there's a lot of different ways that you can regulate. The most common are here at the start of transcription, or whether transcription starts, and then later on here once you've made the product, controlling its activity. All right, so we're going to review the Lacl operon here. I know that you've all learned about this before, so this should be largely review for you. But this is both historically and conceptually important. Historically, the Lacl operon was the first of any genetic regulatory systems to be worked out. And it was worked out in E. coli, and then since then it's served as a model for basic gene regulation. And we're going to get into some more complex examples of gene regulation after we go over the Lacl operon. Okay, so let's just review how the Lacl operon is regulated here. So first of all, the structure of the operon, we have 3 genes. The first one encodes the enzyme beta-galactosidase that cuts lactose, which is a disaccharide, into its 2 constituent monosaccharides, glucose and galactose. The second is LacY, which encodes a transport system to get lactose into the cell, lactose permease. And the third is a gene product called a transacetylase that controls another function, which isn't really essential for Lacl operon function. So we're just going to focus on the first parts of these. Okay, so in terms of regulation, we have a promoter for the Lacl operon. This is where RNA polymerase is going to bind, going to transcribe through these 3 genes, and then of course there will be a terminator here to end the messenger RNA. So this is what the messenger RNA is going to look like. Again, 3 genes on this is a polycistronic operon. Each of those genes is translated independently to make the product proteins. Okay, regulation. Adjacent to the Lacl operon is actually the gene encoding the regulator of the Lacl operon. Lac repressor. So the lactose repressor is capable of binding to the operator region, which is just downstream of the promoter for the Lac operon. And when this is bound here, it prevents RNA polymerase from being able to initiate and elongate transcription to generate the messenger RNA for the operon. Okay, so looking at the mechanism of repression, we're going to go on to the next slide, but these now you have all the basic players. Well, I take that back, you don't have all the basic players, you have the controllers for repression of the system. Okay, so just in general, some terminology here when we're talking about operons. All operons have a promoter, which is where RNA polymerase initiates transcription of the operon. They may have an activator, they may have a repressor, they may have neither of these depending on how they're regulated, right? An activator is a separate protein that will affect the activity of the promoter. Likewise, the repressor is a separate protein that controls the activity of the promoter, all right? So I color-coded these in this version of the slide. You'll have to do this yourself on this copy that you download. But so that red indicates DNA sites, green indicates proteins, and blue indicates small molecules that affect regulation. The other protein that's not shown on here is, of course, a protein complex, RNA polymerase, that we learned about last week. All right. All right. So these are what each of these components are, these systems, right? In the Lac system, the activator is going to be CAP. The repressor is going to be Lacl. The operator for Lacl is going to be downstream of the promoter. The activator binding site is going to be upstream of the promoter. The effector molecules in general are small molecules that control the activity of activators and repressors. In this case, an inducer could be something that either turns on an activator or relieves the repressor. A co-repressor is a molecule that is specifically binding to the repressor to activate it, to try not to use any of these other words here, that the activity of the repressor is dependent on the binding of the co-repressor, right? Now that's not going to be something that we have in the Lac system, okay? All right, so the functions of the different components of the system, I already talked about. Lactose permease is in the cytoplasmic membrane. Lactose is a disaccharide that comes through lactose permease. This is actually a symporter, so this is dependent on the proton motive force to energize it. And then beta-galactosidase cuts the disaccharide into the monosaccharide. There's actually a side activity of beta-galactosidase that produces this molecule called allolactose, which turns out to actually be the inducer, which when it binds to the Lac repressor, pulls it off of the DNA or stabilizes a conformation that doesn't bind as effectively to the DNA, to be more precise. All right, so another term that's used in the Lac system is this concept of catabolite repression. Right. So the way that the CAP comes into this is by virtue of the fact that E. coli would rather eat glucose than lactose, even though lactose has glucose as part of it. The ability to use glucose is constitutive. The ability to use lactose is regulated, and if there is glucose there, then lactose will not be used. Talk about the mechanism of that a little later, but this plays into the regulation of the system as well. This biphasic growth curve is showing that the glucose will be used first, and then the lactose is used. Now, you have to kind of take our words for this a little bit because this is showing a culture that has both glucose and lactose present, and you have to be able to actually measure the amount of glucose being consumed versus lactose consumed in order to actually prove this. But the bottom line is that it doesn't eat both of them at the same time. It preferentially eats glucose and then uses the lactose. If you look at beta-galactosidase activity, you can actually see that if you add glucose to a culture, you'll turn off the expression of beta-galactosidase. Now, I think this graph, this slide is going to be a little confusing, so we'll go over it more in the live lecture. All right. All right, so again, this should be basic review for you. The idea is that there is an operator site for Lacl repressor, that when there is no lactose around, the Lacl repressor binds to this, and RNA polymerase does not transcribe this operon. However, when there is lactose around, allolactose will be produced. Allolactose will bind to the Lacl repressor and stabilize a configuration here that is not does not have high affinity for the promoter. Okay, so you can sort of see these two configurations schematically here. Maybe I'll show you an actual figure of the molecular structure of Lacl repressor bound to the inducer versus not bound, to show the difference between them. But this is a figure from your book, right? So this one has an affinity for the operator, this one does not. So the point is that the Lacl repressor comes off of the operator, and RNA polymerase then is able to effectively transcribe. So you see this messenger RNA here, and of course in bacteria, ribosomes have direct access to the messenger RNA because there's no membrane separating the nucleoid from the cytoplasm, so it starts being translated. Beta-gal is produced by the first gene, permease, Lacl permease by the second, and transacetylase by the third. Okay, so that's what happens when lactose is around. Now, right now, we're ignoring the whole question of glucose. Okay, we're just talking about the role of lactose. Okay, now we get to the role of glucose. So here in this textbook, they call it CRP, which stands for cyclic AMP receptor protein. You've probably also heard it referred to as CAP for cyclic AMP binding protein. The B just gets left out. Anyway, either way, it's the same thing. And the point is that this protein needs another small molecule to bind, in this case, in order for it to bind to the activator site. So unlike the Lacl repressor, CRP, when it is not bound, does not have affinity for DNA. Only when it is bound by its inducer, co-activator here, does it have affinity for the DNA. So that's just the opposite of the Lacl repressor. Now, the other key point here is that the activator binding site is on the other side of the promoter, right? It's drawn here as upstream. I don't know why they drew these slashes here. It's pretty close to the promoter, and in fact, the activator will physically bind to the RNA polymerase to support its ability to bind to the promoter, right? So I don't like this drawing, but this is the drawing they have in your book. Okay, so the bottom line here is that for this particular promoter, RNA polymerase has low affinity for it. Therefore, it needs support from the CAP protein, right? So when CAP or CRP is complexed with cyclic AMP, it binds here and it helps RNA polymerase. It increases its affinity for the promoter, okay? So in the absence of cyclic AMP, you get low transcription because RNA polymerase doesn't bind well here. In the presence of cyclic AMP, you get higher transcription. Now, again, this is ignoring the role of lactose. This is just talking about glucose. Now here we combine the two of them, okay? So we have 4 different possible conditions depending on whether glucose is present or absent, or high or low as shown here, whether lactose is high or low shown here, okay? And that's going to affect the ability of these respective proteins to bind to their operators, their binding sites in the context of the promoter, which then is going to affect whether RNA polymerase is going to be able to initiate transcription. So in order to sort this out, what you have to remember is if the Lacl repressor is bound to its operator, transcription is not going to go anywhere. It's going to stay very low. Okay? Doesn't matter what cap and what cap is doing. If the Lacl repressor blocks the way, then not much is going to happen. Okay? But the only way to get high expression is to have cap bound to cyclic AMP. Okay? So because we have to have the Lacl repressor out of the way, and we have to have the cap bound here with cyclic AMP, the only way we can get high expression here of this operon is to have low levels of glucose so that cyclic AMP will stay low, and high levels of lactose so that the Lacl repressor will be gone. Now that reminds me that what I should have told you here is that cyclic AMP is produced by the cell by an enzyme called adenylate cyclase only when glucose concentrations are very low in the environment. Okay, adenylate cyclase is on there, and so cyclic AMP is produced, right? That's why there's this relationship here. Cyclic AMP is only produced when glucose concentration is low, and then it binds to cap, right? So you don't get cyclic AMP being produced when glucose is high here. Okay? All right. So that actually was somewhat complicated, and I realized that there are some things that aren't in there that should have been in there, which is the role of adenylate cyclase in producing cyclic AMP when glucose levels are low. Then the last thing here is another phenomenon that also affects whether lactose can even induce the system at all, which is that glucose transport. So when you have glucose in the environment, it's transported by this system here. This is what's called a PEP-dependent transport pathway. When glucose is coming in here, one of these components here will actually inhibit the lactose permease. Okay, so that means that if glucose is coming in from the environment, then lactose won't be coming in from the environment. Okay, so that's another way that this, in E. coli cells, induction of expression of the Lacl operon is affected by the presence of glucose in the environment. So you actually have two processes: the activity of adenylate cyclase producing cyclic AMP and the transport of glucose into the cell affecting lactose uptake. Okay, so the transport aspect of this is called inducer exclusion, right? Okay, so that is the lactose system, and it's not as simple as just the Lacl repressor. So you need to make sure that you understand the glucose side of it as well. All right, before I wrap up this first video, I just want to talk about one other system, which is the trip operon. So the trip operon is a biosynthetic operon, unlike the Lacl operon, which is involved in catabolic or breakdown activities. The trip operon is involved in anabolic or building up activities. Okay, so for the trip operon, there's multiple genes here. There's actually 5 genes in this operon: trip, E, D, C, B, and A. We have a promoter here and we have an operator here. Our regulator in this case is the trip repressor. Okay, so when there is not much tryptophan in the cell, the trip repressor does not bind to the DNA. Okay, so tryptophan is a co-repressor here. Okay, Essentially what it's monitoring is if there's a lot of tryptophan around, the cell doesn't want to bother making more, okay? It's only if there's not much tryptophan around that the E. coli cell wants to express this operon and make more. Okay, so the operator is located here in the same position as the Lacl operator that the Lacl repressor binds to. But the key thing here is how this effector molecule works. In the case of tryptophan, when it binds to the trip repressor, it stabilizes the effective DNA binding conformation. So you turn off this operon when you have a lot of tryptophan around. If you don't have a lot of tryptophan around, there's nothing binding here, and it has low affinity for DNA. So the RNA polymerase will hop on here and express this operon in order to make more tryptophan. So again, the role of effector molecules varies between systems as to whether they support DNA binding or oppose DNA binding. Okay, and then the trip operon, this is how it works. There is other regulation of the trip operon, and it has to do with this trip leader, and we're going to get back to that later on. Okay, I'm going to stop this video here, and the next video will have to do with more complex forms of regulation.